Good morning, everybody. Ba-ba-dee-ba-doo. All right, welcome to the last class of the semester, which is kind of crazy. Okay, let's see what we're doing today. So plan today is to finish up material. Ooh, got a bit of a sneak preview there. Oh yeah. Haha, nothing cuter than a fat mouse, right? Although I think that mouse may object to being labeled either cute or fat. All right, so essentially what I want to do with this little section, and this is really a kind of small subset of PowerPoint 20. And so the exam on Friday, I'll just write this down for posterity. Exam 4 will be on what we cover in class, right? And nothing more. So 17, 18, and what is it, slides 11 to, what's the end of this one? 20 something? Pardon me, 21. Of PowerPoint 20. And so for reasons of time and, kind of focusing on the hard but important stuff in PowerPoints 17 and 18, we're not going to do anything from 19. It's lots of cool, fun information, neat information in there. But I just wanted to show you this stuff because it's cool, right? And also I get to show you a fat mouse, which is super cute. Can you imagine having a pet fat mouse? It would be super neat. Anyway, so really what I want to do is just show you some of the couple of, well, I mean, maybe even some more of that, some really cool techniques, right? So one of them is a fairly old by science standards technique, right? It's been around since the early 2000s, right? Which is pretty ancient. Kind of funny, I was doing my PhD then. That's actually before some of you were potentially even born. And also one which has kind of really come to fruition just the last 6 years, I think, 7 years maybe, which is CRISPR-Cas9, which is both a revolutionary, right? In certainly CRISPR-Cas9 is now an RNAi was very much back in the day. And also it means I get to show you this super cool picture. So this is a transgenic alligator embryo. It's going to blow it up a little bit. And I love these kind of microscopy pictures, right? So this is a multicolor microscopy picture. This is with a confocal laser scanning microscope, right? Which is what gives us this beautiful precision, right, and resolution. And this is actually a merge of 3 different color pictures of the same thing, right? So this is a alligator embryo. I'm not really sure what stage, probably kind of like the first third of development, maybe kind of around midpoint, maybe. I'm not super familiar with alligators. But different things are labeled with different fluorophores, right? So the green you can see here, right? This is actually the developing brain. And these green tendrils, those are all neurons, right? And so essentially that line going along here is the developing spinal cord, right? The kind of muscle determined tissue, which is not muscle yet, kind of mesoderm actually, which is the heart here. And also these are what are called somites, right? So these are blocks of tissue that will eventually develop into vertebra and also kind of like the muscles that go into your back and some other bits and pieces, right? And so we have this too, right? So if you did the same to a human being, which you obviously can't, right? Can't make transgenic adults, at least not ethically. You would also see this same arrangement of blocks of tissues called somites. Every vertebrate has them. And basically the number of vertebrae you have is determined by the number of somites that you have. So like alligators, they have loads going down because they've got a tail. We stop around kind of around here, give or take. Rear legs will be coming up from here. And, you know, obviously snake have loads because they're really long and thin and have lots of vertebrae and stuff like that. But this is just awesome. It's such a beautiful picture. It really is. And this actually won an award for, you know, scientific photography, which I think is cool. Okay, so this is obviously beautiful, right? And it was done for a scientific purpose. This was actually to compare development in chickens or chick embryos and alligator embryos because they are evolutionarily related, right? So chickens and alligators, things like that, are somewhat descendants of dinosaurs, kind of in a way is one way of looking at it. It's a little bit more complicated than that, but that's the gist of it. So one of the first things that I want to talk about is RNAi, right? So RNA interference is really an application of, come on, this, right? So what we and other animals use endogenously, right? So naturally to control, you know, the stability and translation of mRNAs, we can actually subvert that in a sense and use it for our own purposes. So normally these are targeted to specific transcripts, the 3' UTRs typically, of specific transcripts to control their expression. But if you were to introduce a piece of double-stranded RNA that matched part of a gene, right, part of a transcript which you wanted to get rid of, you can actually induce this system to knock down gene expression, right? So you don't knock it out, right? This kind of terminology is important. Knockdown means reducing gene expression, basically through reducing translation, right? And so this, and typically it's via mRNA degradation because you get to design this, right? And so you make it a perfect match. And so essentially, whatever gene you target, you get rid of its mRNA, right? Whether it's being, even when it's being expressed, right? And so this is an extraordinarily powerful technique, right? And part of the reason is that you can do this to normal worms, normal organisms. Sorry, I always talk about worms because that's what I work on, right? You can do this at normal organisms, right? As long as you can get this process to work. And so you don't have to mutagenize these organisms. You don't even have to make transgenic organisms, right? You don't have to modify these in any way, at least for C. elegans. Drosophila is a bit different. And so that means that you can take a perfectly normal worm and just screw it up, right? And see what happens. And whatever happens is somehow related to the normal function of whatever gene you're disrupting by RNA interference. And so this allows you to do really cool stuff. Actually, one of the kind of the first big things that came out of this after its discovery in C. elegans, the Nobel Prize, was a researcher in the UK built a library, and she really did shot for the stars, right? Because this was an amazing undertaking. She actually, well, she and her researchers cloned every single C. elegans gene into vectors to produce this, right? So you can, if you have, I think it's around $15,000 now, you know, price of a decent used car, you can actually buy a library of every C. elegans gene. And so you can just knock them down. I don't have one because I don't have 15 grand spare. Whenever I need to do this, I just call up my boss or former boss in Penn State and ask her to get someone to send me them instead. Right, and so you can do this by injection, right, which we don't do in C. elegans because it's very time consuming, but you can. So you can inject these double-stranded RNAs into animals. In worms, you can actually feed those animals that essentially bacteria, you kind of make the bacteria express the double-stranded RNA. And so you just feed them, and then by magic, it's actually not completely understood how this happens. Worms take up that double-stranded RNA and it knocks down gene expression. And in Drosophila, it's even, in some ways, it's kind of a little bit more laborious, but it's even cooler. And so in Drosophila, you actually make transgenic flies that can express this, but only do so under certain conditions, right? So now you can have a normal fly, sort of, flying around, and then you add something to its diet, and you can kill it, or reduce the function of a particular protein that's being expressed by expressing this double-stranded RNA. And you can do that in a tissue-specific manner too, which is also super powerful. So you can knock this gene out in, eyes and not the body, or neurons and not muscle, for example. And so just show you a couple of kind of cool examples, right? So this is in worms. There are lots and lots of pictures of this in worms because it was the first organism this was discovered in, but it's also super, super neat. And so these are a couple of mutants, right? So this is the head of the worm. This is its pharynx, which it uses to suck up bacteria. And this is a mutant called a blister mutant, right? And basically what that means is that its cuticle, this thin layer here, it's essentially the same as our skin, but thicker and tougher, right? And it's what protects worms from its environment, essentially, right? From pathogens and stuff. And in this blistered mutant, this cuticle kind of lifts off. It's basically not being held to the epidermis underneath. And if you knock down this gene by RNAi, this is a TSP, stands for tetraspanin, which is like a kind of like a connective tissue protein. You get exactly the same kind of thing, right? You can see this cuticle is bulging off of the worm here, right? And so you can actually kind of look at this in real time. These are images of live worms, right? So you can follow them through development. You can look at other aspects and stuff like that. And the cool thing is as well is that in humans, there's a disease that's related to intermediate filaments. So these are cytoskeleton proteins. And basically, if you have mutation in one of those genes, you get this really kind of like horrible loose skin phenotype where your skin isn't attached to the dermis underneath, right? So you get these like large kind of patches of skin which aren't stuck to you, which is quite horrible. And so this is one way of kind of studying that, essentially, when you don't have to use people. Kind of basically a way of figuring out some of the basic questions before you get into more complex models. And this is an example in Drosophila. So this is actually comparing the phenotype you get from knocking down the gene versus knocking out the gene, right? So this is essentially a mutation in this gene, these ones here, so TRA2, whatever that is, and these other genes down here, and these produce particular phenotypes. And so what they were doing was saying, well, if we knock down that gene instead of knocking it out, right? So again, this is in a normal background with normal gene function for that gene. Now, if you turn on this RNAi system, do you get the same kind of phenotype? And what they show is that you do. So this is all about patterning, right? So this looks like a male, even though it isn't, right? It should look like so. This is the normal wild type. This is involved in eye development, so you get this vestigial eye. And I'm not entirely sure what this is all about. Something to do with this patterning on its head. I don't know what that's there for. right? So it's a very powerful technique, right? And it really does kind of open up a lot of doors which you probably wouldn't have access to otherwise, right? So if these uh, mutations are lethal, right? And so I think this is a mutant of the same gene here, but you don't see that extreme phenotype, right? So this is likely a less severe allele, right? Where function is reduced somewhat, but not completely. Whereas if you reduce function completely with a mutant, you probably won't get that mutant out of a mutant screen because it will be dead, right? It won't be able to produce progeny. And so that allows RNAi allows you to kind of do a more severe phenotype while still not worrying about kind of maintaining those mutant worms. Makes life a lot easier. Anybody have any questions about this? Time to drink my coffee too before it gets cold. And it's really hard to kind of get over how big of a deal this is. So in terms of mutations, Rogelio, a decent chunk, I think they estimate about a third of genes are essential. So you need them for some aspect of development or reproduction or survival to adulthood, essentially, right? And that's in most organisms. And so if you get a mutagenesis screen is basically you damage the DNA of a whole bunch of normal worms, and then you look for ones which show the phenotype that you're looking for, right? Blistered in this case, or short and dumpy, or whatever, right? And then you kind of work with those mutants so you end up with just the mutation in a single gene, which is presumably something you're interested in. And so for the genes which are essential, if you get a mutation which gets rid of all function, then that worm dies, or that mouse dies, or that fly dies, or doesn't reproduce. And so that allele, that mutation, never gets passed on to the next generation. And so even though you can create lethal mutants temporarily, you can't do anything with them. Right? Well, it's very, very, very hard to. You have to get into things like balance of chromosomes and stuff like that, which just makes my head hurt thinking about it. So most genes are not essential, right? So that you can mutate them, get rid of function, and you'll still get a adult that can reproduce, right? But there are a whole bunch where if you do knock out that function, you don't get any offspring. Now you can actually get mutations which reduce function, right? So these are reduction of function rather than loss of function mutants. And so you can get those in essential genes. Sometimes they're kind of conditional mutants, right? So they only produce the mutant phenotype when you change the temperature, so temperature sensitive. Sometimes they just reduce function a bit, so the worms are kind of, or the animals are kind of sickly, right, but still reproduce. So you can get mutant alleles in essential genes. It's just you can't get loss of function alleles, right, if they're essential. Now, RNAi is completely different, right? You just use a normal genetic background, so all the genes are normal. None of them are mutated, potentially, although you can if you want to do other stuff. But you can still get rid of almost all of the function. Not all of it, right? Because this isn't 100% effective, but almost all of it, right? And so essentially you can look at the role of essential genes without having to worry about those worms dying, right? So if, for example, if there's a gene which is involved early in development, right? So anytime you get a mutant, they never make it to adulthood or the point that you want to study them, right? You can use RNAi after that step in development to see what role that gene has later on, right? That's another way of looking at it too, right? So you can control the timing and you control the severity as well by how much you know, give to the worms or how high you express this in flies. So it's a very powerful tool. Basically, you can kind of stand outside of the animal and you can control how much and where this and when, right, this gene is turned off or knocked down. So it's very kind of very powerful, really. But each has their kind of relative benefits. One of the benefits of mutant alleles is that that mutation is present in all cells and all animals, right? And so you'll always, or should always, get more or less the same kind of phenotype and degree of penetrance. Now, RNAi depends on, you know, how much bacteria that worm eats, you know, how much of that double-stranded RNA gets through the intestine, how much gets into the target cells, and so on, right? And so it's much more variable, right? So that's the downside. It's not as robust, I guess. Reproducible would be another word. Cool questions, though. Okay, so the other piece of cool stuff, and this is give you an idea of how big a deal this is. Normally it takes, oh, you know, 20, 30, 40 years maybe, sometimes, for groundbreaking discoveries to get recognized by the Nobel Prize Committee. This took 5 years, which is almost unheard of. Actually, it is unheard of, right? Typically, you get Nobel Prize winners and they're in their like 50s and 60s or even 70s, right? Because the work that they got the prize for was based on stuff they did much earlier in their career, or they've worked on their whole career. I can't remember her first name, Doudna, Doudna, Jennifer, I think. She and her collaborator in France, who can't remember her name, are in their 40s, I think, if that. You know, so they're relatively young researchers. You know, and this really was a astonishing discovery. And the crazy thing is, this discovery came from kind of acquired immunity in bacteria, right? So essentially, when bacteria, not all of them, but many of them, when they get infected by viruses, particularly DNA viruses, and which happens a lot, they take a little snippet of that DNA viral genome, presumably if they survive, right? And they stick that into their own genome. So anytime they see or they get infected, basically what they do is they express all those little snippets, right, of all of the viruses that they've been infected by over their life and their parents' life. And I don't know, it's kind of hard to talk about bacteria because they don't reproduce sexually, typically. But they take those little snippets and then they compare that to the viral genome. And if there's a match, they cut up the virus, the viral DNA. Right. And so this is really, in its discovery, a mechanism of defense against viruses. And what it does, let me show you a picture, it's a lot easier to describe. And so what we do as scientists, I don't do this because I don't have the equipment to get this into worms, right? But a friend down in Houston who works for a different A&M campus does. And so he uses this to modify his worms for whatever he's studying. And so basically there's 3 parts, right? You have your target sequence, you know, whatever gene that you want to modify or piece of DNA you want to modify. Maybe it's not even a gene. You want to stick something somewhere innocuous, right, where it's not going to affect anything. And you have this thing called a PAM, and this is basically a short DNA sequence that occurs frequently throughout the genome, and that you need one somewhere near there because that's essentially what the Cas9 protein needs. Right, it's not the target, right? It's not specific to the target, but it's just kind of a requirement. And then the other two parts you provide yourself. So you need some Cas9 protein. This is the protein that does stuff. And you need what's called a guide RNA. And so this is a piece of RNA that you make, you design and get it synthesized that recognizes or binds to the target DNA. And so essentially the Cas9 protein, this is almost like the RNA-induced silencing complex from here, right? So the Cas9 uses the guide RNA as a guide, much the same as the RISC complex uses the guide strand to find mRNAs, right? So instead of mRNA, this is all about DNA. And so the guide RNA and Cas9 make a effector complex, and that kind of just goes around scanning the genome until it finds the target DNA. And what it does when it finds it is it causes a double-stranded break. That's the key part of this process. It's this site-specific, right? And you can target this down to the base, right? This is really why this technology is so incredibly powerful, right? This is not random, right? I guess you could make it random, maybe. This is very specific. So if you say, I want to cause a mutation at this point in beta tubulin gene, tappy, 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 tap, design your guide RNA. There are programs that help you do that. Punt it off, get it synthesized, and get it into your organism, and badam, you generate a mutation at exactly the point that you wanted to in exactly the DNA or the gene that you wanted to mutate, right? So this is incredibly powerful, right? Before, we always relied on just essentially the randomness of mutagenesis, right? You know, so if you take a few 100,000 worms and dip them into a nuclear reactor pool, they're going to be really messed up, right? And so you're going to get mutations all over the place. And you hope that some of those mutations are in things you want, right? In genes that you want to discover, right? Even if you don't know what they are. And so it's totally random, and you might just never get them, right? You know, if those are lethal or essential genes. And so now we can basically go, yeah, that one I want to change, right? And so that's what makes this such a scary technology, because now we can do that in any organism. I don't think there's a single organism that this has not worked in. And that includes us, right? So if you want to change a gene involved in disease, now you can do that, right? So potentially you can scan your fertilized embryos for I know, sickle cell disease, beta thalassemia, any other kind of genetic defects, low intelligence, future heart disease, right? They don't do it, Jelani, because that's really unethical. You're talking about permanently genetically modifying humans, right? And so that's like the biggest Pandora's box, right, in the history of science, I think, right? So you the technology is there that you can do this, right? So right now, and if you go through IVF, you can screen embryos, right? So you can reject fertilized embryos that you know, have chromosomal defects, right? And you can even reject them based on particular diseases, right? So congenital diseases that would result in, you know, miscarriage or premature death, for example, right? So that's kind of controversial, but most people are like, yeah, okay, that's not so bad, right? You know, and those uh, rejected embryos kind of they just get discarded basically, or depending on the country, they may get used for scientific research if they're not allowed to develop any further, things like that. And so that's fairly controversial anyway, right? Because each of those embryos has the potential for life, even though it is not uh, an individual at that point, depending on your personal perspective, right? But this goes much further. Now it's going to be the case. Has anybody seen Gattaca? It's a really cool film with, dang it, what's the name of that actress from Kill Bill? One that went around chopping people up with a sword. I can't remember her. Something about Amy as well. There's another one. Anyway, doesn't matter. Really, really cool film if you get to watch it. It's all about eugenics, right? And so we'll get back to the mechanism in a second because this is a nice tangent to kind of explore a little bit. Back in the 20s and 30s, even up to the 60s, actually, the US was a world leader in promotion of eugenics. This is something that's a very hard thing for a lot of Americans to uh, kind of come to terms with, right? And this is basically about selection of better genetics in people, right? So Uma Thurman, that's it. Thank you, Rogelio. Can't believe I couldn't remember her name. Anyway, she's in Gattaca. Really, really cool film. Well worth watching. And it has lots of really cool DNA-based kind of themes. The actual name itself is the four bases of DNA, for example. Yeah, so the US was a world leader in eugenics, right? And there's a lot of programs that would, you know, basically forcibly sterilize women of what was perceived of low moral values or intelligence, right? And many of those ideas, right, and kind of, it's actually really, really disturbing, right? Even the use of Zyklon B, right, which was used in the concentration camps in Nazi Germany, was used in the US, and the Nazis got their ideas from US officials. It was used at low concentrations to uh, sterilize the clothes of Mexican immigrants, right? So people who came over the border to work as agricultural laborers, you know, they were, you know, basically treated as inferior. And so anyway, so we have a long history as a, you know, as a society of, yeah, it's absolutely astonishing, right? They even sprayed DDT, right, which is a really strong insecticide, right, which has been linked to cancer in the face of those laborers as they came over the border, right, to sterilize them because they were thought to carry diseases, right, because they were of an inferior genetic background, which is astonishing, right? Absolutely astonishing. Anyway, that's kind of a bit of a tangent. So this isn't that old, right? But now we have the technology to actually achieve some of those aims of improving the genetic stock of uh, individuals in our population, right? And so this technique would actually allow us to do that. And so it's very scary because it's so powerful, right? Anyway, that's a bit of like a history lesson, and it's one that's worth reading into. It's, you know, it's good to know where you're coming from so you know who you are, I think. You know, that's why I like history so much. Um, but anyway, so essentially, once that double-stranded break has been made in the target DNA, one of two things can happen. It's got to get fixed, right? Because if it doesn't get fixed, that's catastrophic. That's essentially the end of that cell, right? It's a death sentence, basically. right? And so if there is no matching DNA around, right? So it's not in the middle of cell division, for example. Basically, the cell does what I call a Hail Mary repair mechanism. And the reason is it's not very good, right? It's not very accurate. It's actually a, well, crap. We got to stick these ends together, otherwise we're dead, right, chaps? So let's crack at it, right? And it doesn't matter if you screw it up as long as you stick them back together again, right? And that process is more formally known as non-homologous end joining, right? And so the hope is that there are only two ends. If there are four, it's like, well, it could be that and that, could be that and that. I don't know, it could be whatever, right? You can have all kinds of different combinations. And as part of the process, the machinery accidentally introduces mutations. Those can be duplications, right? So a little bit of the sequence gets copied, or deletions, but whatever it is, it introduces mutations which generally create a knockout of that gene, right? So this is great if you want to introduce a non-specific mutation, right? So you get to choose the location of the mutation, but not what the mutation will be. And this was really very much the kind of initial tool in the arsenal, right? However, if you provide some DNA, some donor DNA, which matches either side, right? So there's a bit of DNA that matches this side of the break, bit of DNA that matches this side of the break. That bit of DNA will be used to fix that break via homologous recombination, which is a very accurate, reliable process. But the cell only looks for matches, right? You can stick a reasonably sized chunk of DNA in between those matching pieces. You know, like a GFP reporter, for example, is a great example. Or that match in DNA can include a base change which you decide, right? And so when the homologous recombination fixes that double-stranded break, that match in DNA gets flipped into that break. And so that's how you can insert different genes or different bits of DNA. Or if you want, you can create a very, very, very specific mutation in a gene. So say you wanted to change an amino acid from this to this. All you would need to do is provide the donor DNA, which has the like the base change, which will cause that change in amino acid, and that will get essentially replace the old sequence. And so this allows you to do very, very specific mutations and insertions and all kinds of stuff. And so this was actually used by a Chinese researcher or a researcher in China, which is the only time that we're aware of that CRISPR-Cas9 has been used to genetically modify a human. And then thankfully, there's a huge uproar and outrage about this, and which is reassuring, right? Because you don't want it to happen again. But basically, this scientist, what he did was he introduced a deletion in a cell surface receptor called CCR5, which is used by HIV to access or get into T cells in your immune system, which is how AIDS develops essentially. Right. And so if you have that mutation in that receptor, you can't get AIDS. Right. And so he thought, okay, well, this would be a great idea. These kids, they're never going to get AIDS now. Right. Even though those children were only embryos when he did this, and they certainly didn't have any say in that change. And their offspring will get a copy of that change, and they certainly didn't have any say in it either, right? So this is a permanent change to the genome of those two individuals, right? And we also don't actually know what introducing that mutation does for other diseases, right? So there's potential risk that it will cause problems with other diseases. I think smallpox and chicken pox, right? You have less resistance to those genes if you have that mutation. And also you can get off-target effects, right? So this, although this is pretty reliable, right? And the guide RNA generally gets the Cas9 protein to cut the DNA where you want it to, you can actually get off-target effects. So there is a possibility that when you do this, you'll generate mutations elsewhere in the genome which you don't know about, right? And so that's another, aside from the whole ethics of changing a human individual by manipulating their genome, right, which is really dodgy, right? I think there's lots and lots of reasons why you'd want to be careful about doing that. You can also introduce other changes elsewhere that you're not aware of in things which you don't know what their function is, right? Or you won't see that issue until much later in development or even adulthood, right? So it's kind of like with great power comes great responsibility, right? Whoever said that, right? This is truly the power over our own genetic future if we choose to use it, right? And so would you really want that to be used? And also don't forget too, this would be as a, and actually Gattaca is a very good film to address this. Basically, this is something that only wealthy people would be able to afford, at least initially. And so if you were able to genetically improve your offspring and hence their offspring, this is something that wouldn't be available to everybody. This is something that would be available only to the top strata of society, which there's enough issues with inequality as there is without adding an extra one, a more permanent one too, right? Not just wealth, but genetics as well. Yeah, it would cause it's, and also as well, you know, a lot of things which people want to modify, right? Sporting prowess is a popular one. Everybody wants their kids to be great at athletics for various reasons, or intelligence, or health, right? We don't actually know how all of those big things are controlled, right? What are the various interactions? As you've seen already in the amazing complexity of control of eukaryotic gene expression, changing one thing one place can have a very different effect or unintended effect somewhere else, right? So not only would it, you know, potentially create, you know, even more inequality, but also you'd be creating things that you don't really understand until much later, right? You can't change them. Once you've changed that DNA is now in the population, right? You can't kind of close that Pandora's box and stuff all of that stuff back inside it. So anyway, it's a really cool kind of thing to think about, right? You know, this isn't just science anymore. This is also society too, right? And you can't really, at the end of the day, you can't take science out of society. You can't put it separate, right? Because whatever we do as scientists, right, has an impact on society too, and vice versa. To be honest, generally, I don't know, right? That's kind of the scary thing, right? Because it's already happened in a country which has somewhat lax control and regulations over what scientists do, right? Which also has other kind of perverse incentives for scientists, right? So in China, if you publish a paper, you get paid a bonus for it, right? So you can imagine, you know, this researcher was thinking, well, if I get this out, I'm going to get my work into, you know, Nature or Science. I'm going to get $100,000 bonus. You know, I'm going to get tenure and have job security. So that's an issue. But also, you're right. You know, once this technology becomes sufficiently mainstream, there's nothing really to stop, you know, IVF clinics, you know, in Brazil or, you know, Thailand or whatever, to basically add this to their list of, you know, options that you choose when you start treatment. So I don't know. It's a good question. We'll find out as time goes on, I guess. Yeah, designer babies. Thing is, though, human genetics isn't simple, right? So you might be designing something that you don't actually intend. You know, so for example, if you go for, you know, increased muscle mass, you know, you change expression of testosterone, right? Maybe in your male offspring, you might also get increased aggression as a result, right? You might end up designing arseholes, not actually the the object, but the individual. And so it's you know, I don't think human DNA is something that we really want to experiment with, to be honest. You know, because again, you can't undo those changes once they've been made. They will be out there. You can't unmake the mistakes. Anyway, so that's pretty much it from me, to be honest. Which kind of coming to the end of the lecture. I had intended there to be time for questions and stuff in general, but if you have any of those, feel free to email me. I'm more than happy to answer them kind of outside of class too. I just wanted to spend time on this because this is really cool and it kind of really, in a way, brings together a lot of what we've been learning about over the course of the semester, right? This is really very much bleeding edge, right? And it's also, the closer to that edge you get, like the kind of sometimes the scarier the questions become. So that's it from me. Does anybody have any questions in the last couple of minutes? No coffee left. That's it. No more lectures. This is the last one. Unless you want to take this course again, you know, just for fun. Hear me do this all over again for another semester. No, that's it. So the last exam is on Friday. It's not a final in the conventional sense. It's a, oh, no, please, Rogelio, please pass the course, move on to other cool stuff. There's plenty of other cool biology to study. Yeah, so the last exam, which will be on the PowerPoints that I wrote in the chat earlier on in class, be on Friday, standard from 9 a.m. to midnight. You'll have an hour and a quarter. Don't rely on the ability to look stuff up. You probably won't have time to look up everything that you want to look up, right? So study the details, study the concepts, just the same as for exam 3. And there you go. Any questions between now and then, feel free to ask. I'll be in lecture for at least the first half hour on Friday. If you have any last minute questions or issues with the exam. And there you go. That's it. It's always kind of sad when it gets to this point. It's like you're happy because you know you got some time off, but you're also sad because that's it until next time. All right. So have fun, everybody. Stay safe, work hard, and I'll see you soon.